Good evening, everyone. Welcome to Botany Insider's new class. We'll be discussing about the cell cycle in detail. Okay, let's wait for others to join in and then we'll start off with our class. It's a very, very important concept if you look into the previous years. So, let's wait for others to join and then we'll start off with our class. Okay. Please confirm whether the audio and the video is clear to everyone or not in the chat section. We'll be discussing cell cycle in detail. Okay, so be there in the class for the entire session. You'll get to learn a lot. Good evening, Sonam. Good evening, everyone. Good evening, Komal. Okay, so let's begin with our class now. Others would join in the time being. Okay, now first thing first, we have two courses running for the December 2023 students. One is a full course and one is a crash course. Okay. If you wish to enroll in any of these, you can download our application and get yourself enrolled. We have our own application which is available on your iOS as well as your Android application. Okay. So if you wish to enroll, you can download the application and get yourself enrolled in the same. In addition to that, we have our notes booklet as well. You can WhatsApp on this particular number to get the updates, to get the final sample for the same thing. Okay, let's begin with our class now. In addition, we also are about to start a previous year question detailed course for you all. So if you wish, you can enroll. Like we're about to start the enrollments for the same. So you can download our application further and enroll as in when we start off with the enrollments. Okay, let's begin with our class now. We'll be discussing about cell cycle in detail. Okay, talking about this particular topic, first thing first, it's a very, very important concept if you look into the previous years. Okay, every single year, at least one question can be like expected from this particular topic. Now, cell cycle has majorly the two main components. One is the various stages that are there in the entire cell cycle. And second is your various transitions, or we can say the various or the complete concept of regulation of the cell cycle. From regulation every year, like in last 7 to 8 years, every single year question has been asked, and from the stages as well. It's a very, very, like you can easily expect a question from here. Okay, let's begin. Now, first of all, make sure that you're there in the entire class because I'll be explaining you a number of things. So be there in the entire class because you'll be getting to learn a number of pointers. Okay. So just concentrate and just understand the entire scenario. When you look into cell cycle, what is it all about? If I want to define it in the simplest manner possible, what is it? It is a sequence of event. In this, what happens? You have a number of events. So we call it's a sequence of, or we can say it's a series of events that occurs. A series of events that occurs because of which a cell divides into two. Okay. Now, depending upon the cell, we have either the mitosis or the meiosis. Now, if you look into the type of the cell that we have in our body, first type is your somatic cells, and second are your autonomous cells. The sex cells. The first are the somatic cells and the second one are the sex cells. So somatic cells are the ones which are or which just divide into two of its similar type. That is known as your mitosis. Okay. Then you have sex cells. So sex cell undergoes a specific phenomena known as meiosis, in which there is reduction. Like in this, or meiosis is also known as your reductional division. Why? Because you'll see a reduction in the number of the chromosomes. You'll see a reduction in the number of the chromosome from the mother cell to the parent, like from the mother cell to the daughter cells that are getting formed. In case of your mitosis, there is an equational division occurring. What do you mean by equational division? So equational division is that you have one cell, and in the next cell, in your next generation also, you will see equal number of chromosomes. For example, if the cell is 2n, in case of, and it is undergoing mitosis, so your daughter cell will also be 2n. What is n over here, or what do you mean by 2n or n? So 2n and n showcases your diploid and your haploid condition, right? I'm expecting that you all know what diploid and haploid condition is, because it's very basic. Then in case of your meiosis, the initial daughter cell is 2n. Oh, the initial parental cell or the mother cell is 2n. But when we have the next generation formed, you have n in the next generation. That is, it becomes haploid. Why? What is the reason? What are the complete various scenarios happening? We'll be understanding in this complete class. Okay. Now understand. When you look into the various events or the various stages of the cell cycle, you majorly have three stages in the, or you majorly have, like, the broad one are the two stages. Okay. Which are the two stages? One of the stages known as your interphase, and the other one is known as your M phase. Okay. One is known as your interphase and the other one is known as your M phase. Yeah. The other one is known as your M phase. Okay. Now, in case of your interphase, you have three of the stages occurring in the interphase, which is known as your G1 phase, S phase, and G2 phase. The G1 phase is also known as your gap 1. G2 is also known as your gap 2. And S is known as your synthesis phase, right? In case of your M phase, now this M phase is either mitotic or it can be meiotic. Okay, depending upon which cell do we have, we'll be having either mitosis or meiosis. What is it? Like, why is a cell having a different type of division going on? The main reason is their basic nature, and second is the requirement. What requirement is there? We'll understand. Wait. In the M phase, you majorly have four of the substages. Okay, what are the four substages? You have prophase, you have metaphase, you have anaphase, and then you have telophase. Okay. Prophase, metaphase, anaphase, and telophase. Now, these four stages, pro, meta, ana, telo, all these four stages occur in your mitosis as well, and it occurs in your meiosis as well. What is the major difference between the two? Understand. In case of your mitosis, you have prophase, metaphase, anaphase, and telophase. In case of your meiosis, you have meiosis 1 and you have meiosis 2. Meiosis 1 have prophase 1, metaphase 1, anaphase 1, and telophase 1. Meiosis 2 have prophase 2, metaphase 2, anaphase 2, and your telophase 2. Right? Now, what is the main difference occurring? Understand this, very, very important. Now, the main difference is that in case of your meiotic division or in case of your meiosis, in the meiosis 1 and in the prophase 1, you have 5 more stages occurring. What are the names of the 5 more stages? You have leptotene, zygotene, diplotene, dikinesis, right? What are the various four stages? You have leptotene, you have leptotene, you have zygotene, you have pachytene, then you have diplotene, and then you have diakinesis, right? What are the five stages? You have leptotene, zygotene, pachytene, diplotene, diakinesis. Right? Now, why are these stages occurring? Understand this again. Very important. See, in case of your meiosis, we have the reductional division, that is 2n getting converted into n. And this particular division occurs in your sex cells. Sex cells are your gametes. These are your gonads, right? Male sex cell and the female sex cell, these two have to fuse together to make a zygote, and then the new generation will be formed. So there need to be some kind of variation, because we, like, these are sex cells, there need to be reductional division, there need to be, like, after a point there need to be, like, to maintain the zygosity, to maintain the entire, like, ploidy level, we can say, because in meiosis these sex cells will be, like, gametes will be formed. If imagine a scenario, male is 2n, female is 2n, in the next generation you will see 4n, which is not, like, it's not viable in nature. So to maintain the polyploidy, to maintain the ploidy level in the entire species level, so you need to make sure that when the male and the female are fusing with each other, they are having half of the number, which occurs because of meiosis. And in meiosis, why do we see variation in the nature? Why do we see the two species that are there, the two individual in the same species that are there? Why are they different from each other? They are different because of a phenomena that occurs in the Pachytene stage of your meiosis 1 of your meiotic division. What is the phenomena? The phenomena is known as crossing over. What is the phenomena? Phenomena is crossing over, which is causing genetic in a different class. Okay. So you have Pachytene stage in which crossing over occurs, which causes genetic recombination, which leads to the variation in the individuals. If genetic recombination is not there, there will not be any kind of variation in the organisms, and as a result, every single organism on the earth, let's say of human race, they all will look similar, because if by chance there is no genetic recombination, how will you see the variation? How will you see the various different type or the characteristic features? See, we all have the same body structure. Everyone have two limbs, everyone have like two hands, two legs, one nose, two eyes, etc. But everyone have a different shape, a different structure, a different orientation. Orientation in sense of some have bigger eyes, some have smaller eyes, right? So this is because of the variation. This is because of the genetic recombination which is occurring, right? So that's the reason why you have the Pachytene stage, and this Pachytene stage causes the, or in this Pachytene stage you have the crossing over. This has been asked a number of times in the examination, so remember about this. Okay, now what happens in the M phase? You have either the meiosis or the mitosis, whatever is the type of cell it occurs. Now in this also, at this point there is division of the, like, the cytoplasmic content, or we can say initially you have the content getting, like, separated, right? But the actual division of the cytoplasm, that occurs after you get done with the content separation. What is the name, or what is the process of the division of the cytoplasm? Can anyone write down in the chat section? Yes. if the cytoplasmic content is now like, now you have to make, you have made the two cells, all the content is there, every single thing is there. So what is the process of the division of the two cells getting separated from each other? Is it karyokinesis or is it cytokinesis? Everyone, think about it. Try to write down in the chat section. What is it? Karyokinesis or cytokinesis? Very good. Yes, it is known as your cytokinesis, right? So, what is it? First, you have the karyokinesis occurring, and then after karyokinesis, you have the cytokinesis occurring. Okay. Karyokinesis, the word karyo, this is related to nucleus. Okay. So initially your nuclear content is getting separated. So for that, or for this nuclear content to get separated, the karyokinesis word is used. For the cytoplasm to get separated, the word cytokinesis is used. Okay. Remember about this particular thing. So in the M phase, there is karyokinesis occurring. After this, you have the cytokinesis occurring. Okay. So now in your G1 phase, like, this interphase is actually the main preparatory phase, the main phase in which the cell is getting ready for the division. Okay. Gap 1 phase is the one in which the the cell will enlarge in size, and it will make sure that all the proteins that are required for the synthesis of your DNA. Remember, it is the S phase in which your DNA synthesis occurs. Very, very important. Okay. The DNA synthesis occurs, for which you require proteins. These proteins are subjected, or these proteins are related. The proteins that are required in your DNA synthesis, they are produced in your G1 phase, right? And then in the G2 phase, you have a number of other processes occurring, so that the cell can then enter into the M phase, which is your either mitotic or meiotic phase. After that, you have the prophase, metaphase. It's the phase in which the chromosomes align on the equatorial plate. Then you have anaphase, in which the sister chromatids move to the separate poles, to the opposite poles. Telophase, now your sister chromatids, or they get condensed into your chromatin fiber, and then the further process continues. Okay, so this is the complete gist of what cell cycle is all about. There are a number of different things that you have to keep in mind for all of these. We'll be discussing about that as well. Before that, just go through it. If you find any difficulty, any problem, you can ask. Okay, I have tried to explain this particular thing to you in the simplest and in the most slow manner possible. Check if you understand this. If yes, then we'll proceed further. Okay. Quickly go through this, everyone. You can ask your doubt if any. Think about it. Like, if you find any difficulty at any point, you're free to ask. Okay. Understood, everyone? Any doubt? Any problem? There are 19 students in the class. I'm expecting this particular thing is first clear. Chiasma formation, I've not yet explained. I've not yet explained that. I've just explained you the base content for the same. I've not yet gone into the depth. Ask your doubts according to what I have explained. Whatever I've explained till now, ask your doubts from there, if any. Clear? Any other doubt, anyone? Done? Okay, let's move forward. Understand this now. So, what is cell cycle all about? So, it's a series of events that take place in a cell that leads to the division and the production of two daughter cells, right? It's a very important process that ensures proper growth, proper development, proper maintenance of the organism. Four main stages: S, G1, S, G2, and N. Okay. G1 phase is the one. During this phase, the cell grows in the size. It synthesizes a number of proteins so that the DNA replication can occur in the next time, in the next particular phase. Then you have the G2 phase, in which, like in the S phase, the major thing is that you have the formation of your DNA, or we can say the DNA replication occurs. I'm expecting that, like those of you who are studying or who are having a self preparation done, I hope cell the DNA replication concept is clear to each one of you, because it's a very important concept. Okay. Every single year you can find a question. In 2022 also there was a question asked in which you were given a very simple diagram and you were asked about the directionality of the entire thing. In 2022, in the morning shift and in the evening shift, the questions, like in one of them, the question was related to the directionality, and in the other shift, the question was related to, like you were given a diagram again and you were asked at which particular place will you find the DNA, where will the Okazaki fragment will be formed at. There were four of the options given. Okay. So make sure that you are doing every single topic, that you're doing it from like you're doing every single topic from in the best manner possible. Okay. Because it's a very, very important concept. Okay. Make sure that you understand this. Okay. I hope it's clear. Then you have the G2 phase. In the G2 phase, the cell continues to grow and prepares for the cell division. It synthesizes necessary proteins and the organelles that are required for the division, and the cell checks for errors and all of these things. Okay. Is it clear till this particular point, everyone? Just check. Yes, Ayush, I am from Deen Dayal Upadhyay College. You're from which batch? Yes. Ayush, which batch? I passed out in 2017. You're from which batch? Yes. Everyone just go through all the theory pointers. Necessary proteins like MCM helicase, necessary proteins like like when we discuss about the DNA replication, you have seen a number of proteins are there. For example, CDC6, for example, CDT, for example, MCM helicase. So all these proteins need to be synthesized, right? For your DNA replication. Then for the formation, or for the proteins to get separated away from each other, there is requirement of tubulin, right? For which is required for the formation of the spindle fibers, right? We have discussed about all of these things, Vagisha, in the class. You are from crash course, right? We have discussed all these things. Clear everyone till here? Let's move forward, right? Now see over here, you have prophase, metaphase, anaphase, and telophase. Prophase is what in this the chromatin condenses and the duplicated chromosome becomes visible. Then you have the nuclear envelope disintegrating and the mitotic spindle begins to form. You have to remember that at which particular stage, what is the change occurring, what is the new type of, like, what is the functionality going on. Reason being, these changes are important for you to remember, and in the examination you get questions from these things, right? Anjali, wait for a minute. Let me cover this, then I'll answer your question. Okay. Then you have metaphase. In the metaphase, you have the spindle fiber that attach to the centromeres of the chromosomes, aligning the chromosome at the equator. Then you have anaphase. So in the anaphase, what do we have? The spindle fiber shortens and then they separate the sister chromatids. Last is the telophase. They'll be getting separated or they'll be moving towards the opposite pole. Over there, the chromosome will start to condense and you'll be seeing the cytokinesis going on after the complete disintegration. Okay, these are all the pointers. I hope the basics of the complete cell cycle is clear to all. Okay, now Anjali, coming to your question, G naught phase is also known as quiescent stage, or we can also call it a resting stage. So when the cell, if either by choice wants to quit the cell cycle and rest for some time, it can rest, maybe because of lack of some proteins, maybe because of lack of some of the energy first, and second, it can exit the cell cycle if by chance that cell is not required to divide at that particular time, and it gets a signal to just quit the cell cycle and rest till the time the body does not require. Okay, clear now, Anjali? Understood? Okay. Two type of spindle fiber. There are various proteins that are there which makes up the spindle fiber. Okay, we'll be discussing about that in the next class. Like, we'll be discussing in detail about that as well if we schedule a class in detail on YouTube. And if you want to learn in depth, you can come in our full course or our crash course. Okay, we're about to start a second cycle of our revision, so those of you who are interested, you can join in that particular thing. uh, Textile and interzonal, I have not heard about this thing. I'll read more about it and I'll come back to you. Okay, and I'll get back to you for this particular point. I have not yet heard about these two things, interzonal and textile. Okay, so this is about for this particular class. I hope it was helpful. Now you can let me know in the chat section which are the other topics you want me to take up, because it helps me to schedule the classes for the next week easily. Okay, just let me know which classes, which topics, or what type of classes, whether it's your question topics only or only the theory topics, what type of topics do you want? Yes, quickly. And was the class helpful, everyone? Those of you who have joined, was the class helpful? Yes, cell signaling. Okay. We have already covered few topics of cell signaling. You can check that. We have covered GPCR. We have covered IP3 DAC pathway. You can check out those things. Okay. So this is about it. Let's meet in the next class. All the best. Good luck, everyone. Just a reminder, if you are looking for a structured course, we have a full course and a crash course running. You can enroll in our crash course. We are about to start like a complete new cycle of the entire syllabus revision. In addition, you'll be getting PYQ course, like the complete PYQ discussion that we are going to start in this particular course. You'll get in this course or in this course for free. Okay, so you can download and you can get the benefit of two courses together. So this was about it. Thank you for joining in. Bye everyone. Take care. All the best.